Hello everyone. In this video, we will be discussing about the DNA polymerase, a brief introduction about the DNA polymerase. We will first see about the prokaryotes. So it was basically the author Kornberg actually who characterized and purified the DNA polymerase from the E. coli cells. And we know now that this DNA polymerase, this is actually a single polypeptide enzyme, and we call it as DNA pol 1. So this is encoded by the gene pol A. Now, moving forward, we know that the structure of the DNA polymerase is like it contains the palm region, it contains the thumb region, and the fingers. So here, this is the DNA that gets bound to the thumb region. Now we can see here in this image, so we have here the DNA and we have here this insertion site basically. So this is the insertion site and this is the palm region and this is the thumb region and these are the fingers and this is the post insertion site. Now we will see here how exactly the incoming nucleotide bind to the DNA polymerase. So we know that the fundamental reaction that is done here is the phosphoryl group transfer, and here the nucleophile is the 3' OH group of the nucleotide at the 3' end of the growing strand. So we can see here that, for example, we have here this incoming nucleotide. So here this is the incoming nucleotide. Here we have, this is the template strand. So this is the primer. So it provides us the 3' OH group. So here we have in the DNA polymerase the two magnesium ions that bind at its catalytic site. So first is here and second is here. So in the first catalytic site, for example, when the Mg2 positive bind is there, so what it exactly does, so it helps to deprotonate the 3' OH group. So here we have the 3' OH group, so it will try to deprotonate the 3' OH group and making it more effective nucleophile. And then we have here the second magnesium ion. So what will it do? It binds to this incoming nucleotide or incoming DNTP and it will facilitate the departure of the pyrophosphate. So here the pyrophosphate will get released. So this provides the departure of the pyrophosphate. So pyrophosphate will get released. Now here we can see now further, we have here the primer strand, then we have here this template strand. So this is here the post insertion site and this is the insertion site and this is actually the incoming DNTP. So here we can also see that now the translocation will happen. So here is the translocation. So here the translocation will happen now. So this was all about this topic. So subscribe our channel and stay connected.